and I ask you to work with Salmonella typhi. Salmonella typhi is a class 2 pathogen, risk group 2 pathogen. So at the biosafety level, you need to work.
Very famous or very in trend. First, because there are a rapid advancement in immunology. So yeah, we need to be aware that there are the explosion of knowledge in immunology. Many research have been done. There are many high-end equipment like mass spectrometry and everything that can get a very high throughput data. You can get very big data. So data is a lot, right? Advancement in high throughput technology has generated vast amount of immunological data. And then this data is not simple, it's very complex. So immunological data, including genomic, proteomic, and structural information, is usually complex and very challenging to analyze using traditional methods. So there come the needs for computational methods. Okay? Immunoinformatics field provide computational tools and methodologies to process, analyze, and derive meaningful insights from large-scale immunological data sets. So how do we apply, or to what field do we apply this immunoinformatics? First, in vaccine design. Immunoinformatics plays a very crucial role in predicting antigenic epitopes, optimizing vaccine candidates, and accelerating vaccine development process. Okay. Second is in the field of immunotherapy, where you target immune cells usually to for the therapy of certain disease like cancer. So by identifying potential targets for immunotherapy and predicting immune response, immunoinformatics aids in the development of novel immunotherapeutic strategies for cancer, autoimmune disease, and infectious disease. And third is in drug discovery. Computational approach in immunoinformatics facilitate the discovery of small molecule drugs, biologics, and immunomodulatory agents by targeting specific immune system component pathway. Right, but there are also some challenges as well as opportunities in immunoinformatics. Like for example, the data integration. So because the data is often a lot, we have diverse type of data such as genomics, transcriptomics, proteomics. So it's very challenging in immunoinformatics, but it also presents opportunities for like comprehensive analysis. But we really need to be very proficient in analyzing analyzing this data. Okay, and then next, before we go further into this discussion, I think it's maybe worthy to revisit immunology, right? So what is immunology? It is the study of the immune system, which functions to protect the body from pathogens and other foreign substances. It involves the investigation of cells, tissues, organs, and molecules that play a role in immune response. It seeks to understand the mechanism by which the immune system recognizes and responds to infectious agents and other foreign substances. So in this world, outside our environment, we have so many what we call allergens. So allergens is the substance that can induce reaction, any reaction, and all of us are unique, and each of us has a set of a very specific immune system. So maybe I'm allergic to peanuts, but not you. So it's how our body reacts to the certain allergens. But usually the immune profile for the certain population, community is not the same, but it's quite similar within the same population compared to another population in a different geography. Because I remember when I further my study in the UK, they have a season of flu fever. I can't remember, but many of my friends, my European friends, they are allergic to what we call it? The bunga, I don't know what it's called. Pollen, like pollen. So In the antibody, so antibody will come to the antigen. So this is your antigen. The specific surface on the antigen that to the antibody, and specific surface on the antibody. So this is the epitope part. This is the paratope part. This one the same like previously. Oh, they just want to show the epitope part. So there are two types of response, humoral and cellular. In humoral, the most important guy is antibody. In T cells, there are no antibody in T cells. The most important guy in T cells is the cytotoxic T cells as well as T helper cell. Okay. So the molecule that has the epitope prediction part we call it MK. C is the constant region, V is the variable region. It's this chain a variable region, hinge region. The hinge region is very flexible to allow for antibody and antigen to hinge region. There are two types of epitopes, linear as well as conforming or linear. We also call it sequential, or we also call it continuous. B and this is antibody. It is your antigen that binds to we call it information or because it's non-linear, discrete amino acid sequence come together due to cloning, and their size is usually between 15 to 22 properties of your structural feature of pathogen molecules, and then there are molecular interactions between paratope and the interacting region of its target molecule. B cells neutralize pathogenic molecules by targeting them with antibody. Okay. So this is just the example binding of a discontinuous epitope. So you can see that here is the example of antibody fragment complex with linear foul lysozyme. Okay, the blue one is the epitope. Cannot really see here, but this is the example of discontinuous. Okay, how do we identify B cell epitopes? So there usually previously the approach is always experimental. You need to go to the lab, you need to perform experiment to identify the epitope of each antigen or the antigen of your choice. Right? For example, protein crystallography, ELISA, and peptide chips. Okay. But the problem with using experimental method, it is usually expensive, right? If you want to do protein crystallography, do you know how protein crystallography works? Okay. You need to produce the protein and then crystallize. Yeah. So in order to crystallize a protein, you need a very high concentration of that protein first. So yeah, protein molecular work, or like protein cloning and then produce protein. When you have the protein, you need to purify it and then crystallize. When you have this crystal, it's not in the lab anymore. You need to do some physics work in which you use X-ray to bombard your crystal and get the X-ray diffraction, blah blah blah. So this is usually using facilities we call spectrum. There's so much money and expertise will be involved in crystallography. ELISA, it's pretty much lab work, but it is usually a lot of false positive. And also you cannot really, they usually don't provide the structural information. You just know they are okay, they are binding because you are testing binding, but you don't know the exact structural information. Okay, as well as peptide chain. Yeah, so overall they are expensive or time consuming, low throughput, or have low accuracy. Okay, so computational methods have been developed to assist or substitute the experimental approach. So this is a few tools, database tools that can be used to predict the epitope. IEDB, BCI-PAC, Los Alamos HIV database. This is even though the database for HIV, but HIV is an immune deficiency disease, so there are a lot of information about these immune cells as well as protein data bank. But you also need to bear in mind. Now I can say now that computational methods is easier than experimental method. But in order for us, I mean you guys today, to be able to use computational method, previous scientists have we contribute a lot to this computational, because if you understand all these computational methods, they are based on algorithm or based on what we call it language programming. Programming. Yeah, it's a programming. No, we need a lot of data. I words exactly. So it's something that you need a lot of previous data, so the system or any bioinformatics tool can be trained. We call the term train in bioinformatics. So let's say I'm the bioinformatics tool, so I cannot just give you any idea or any suggestion with zero knowledge. So I really need to be trained with previous data, with a lot of data previously done by a lot of researchers in a lot of decades ago. So actually computational methods is also contributed by a lot of experimental data done previously from previous lectures. So only because of that vast of data, because we have a lot of data, so maybe because there are a lot of data, so one bioinformatics tool can be trained, meaning that they know, oh, A is equal to B, B is equal to C, blah, blah, blah. So they can be trained, and then they can propose a new suggestion or a new prediction, or they can predict something based on their knowledge from that data. Right? So I would like to say, without experimental method, there will be no computation. There are two types of B-cell prediction tools: sequence-based method and structure-based methods. Okay? As its name applied, sequence-based method is a prediction for linear epitopes. Right? So there are a lot of algorithms, not only one. It depends on the website, the tools. And they are also using a different characteristic of the protein to predict the epitope. Okay? For example, the first one is protein hydrophobicity algorithm. So this prediction is based on the protein hydrophobicity and hydrophilicity. Okay. The first one predicts epitopes based on the hydrophobicity and hydrophilicity of the amino acid in the protein sequence. Right. So in the previous slide, I already told you that epitopes are often hydrophilic because they are on the surface. So you need to remember the protein that are inside or in the core, they are more hydrophobic because they hate water. So protein in the surface are more hydrophilic. So because epitope they are on the surface, usually they are more hydrophilic. And the second one is protein flexibility prediction algorithm. There are also some algorithms, for example, K+ and Schutz, that predicts epitopes based on the flexibility of the protein structure. Because in previous slide, I have also told you that epitopes are often located in the flexible regions of the proteins. Right? It needs to be flexible in the flexible region, so it can be easily change the shape to interact with the antibody. So when the protein bind to one another, this is the shape. When they bind, they maybe need some conformational changes to really bind like this. So this is the previous shape. When they bind, they change a little bit to bind. So yeah, epitope needs to be flexible to do that. And the third one is protein secondary structure prediction algorithm. For example, GOR2 method. Okay, this algorithm they predicts epitopes based on the protein secondary structure, which includes, do you remember protein have 4 structure: primary, secondary, tertiary, and quaternary. Okay. So this one they predict based on secondary structure, which is alpha helix and beta helix. Okay. Because epitopes are often located in the loop regions between secondary structure. So you have your alpha helix, you have your second alpha helix. Usually epitopes are the loop between secondary structure. And the third, so the last algorithm is protein antigenicity. Protein antigenicity. This algorithm predicts epitopes based on the likelihood that a particular sequence of amino acid will protein antigenicity. Based on the likelihood that a particular sequence of amino acid will give or will induce, or will show an immune response. So that's the algorithm. It depends on the website we use. So this one just now is the first one, protein hydrophobicity. So actually this slide after this is just showing how the algorithm predict the epitope. So it's maybe quite confusing. We as a scientist, like myself, I sometimes also don't know how they decide. We just insert in sequence and I just want the result. This one, okay, give me the prediction. But this is actually how they give you the prediction. If you do the proposal. Okay? So this is the Parker hydrophilicity scale. The values describe the hydrophilicity of each amino acid. So each amino acid has already a score. We call it score. Okay. From the most hydrophobic, the more negative number is more hydrophobic. Up is more hydrophilic. Okay. So based on the sequence, so they will when you insert your sequence, they will give a score to your sequence based on this. It's fixed. This score number is fixed. Okay. Like for example, you give a sequence of FVDEKRB. Or like for example, you give a sequence of full bacterial sequence, and from that bacterial sequence, they will identify epitopes based on the scales. So this is a calculation. So you can see F is minus 2.78. L here, F here, sorry, F minus 2.78. So this is this number they calculate. The average number, you have 1, 2, 3, 4, 5, 6, 7. You have 7 amino acid. So you just calculate all 7 score and divide to 7. So you get 0.3. And from that one as well, you need to calculate the sensitivity and specificity of your prediction. So usually to ensure the sensitivity and specificity, you need to validate the epitope predicted by software. So you need to validate with some experimental or comparison with other software. So usually any methods, not only in immunoinformatics, any method that you use bioinformatic tools, usually you need to take that result, but with caution. Usually you need to go back to the lab and validate with some kind of experimental approach as well. So don't trust bioinformatics 100%. Always, because it is always prediction. So the most famous, I think more famous than immunoinformatics, it's not many people who do immunoinformatics, but there are a lot of people doing protein structure prediction. If you like, protein structure prediction using like homology modeling, any modeling stuff to structure the protein, to predict the structure of the protein. But always compare with other tools or the most preferred is experimental. Always validate some kind of experimental data. Always back up if wet lab. Right, this is to evaluate the performance. Yeah, this is the curve to evaluate the performance of the predicted epitope. This population something. So this is again just a method behind the scene on how the program or how the platform, the tools, the database predict the epitopes from the sequence that you work with. So it can be based on binding motif or matrix, we call it quantitative matrix. It can be based from statistical method, we call hidden cutoff model, or it can be based from machine learning method. Right? So this is the example of the website, the tools. This is free tool. The first one we call it ABC prep. Yeah, we as a scientist we just submit the whole sequence of your, I don't know, maybe bacteria or virus. So you just submit the sequence of the virus, and then maybe put some information about that virus, and then these tools can predict the epitope, which from the whole sequence is the epitope. B cells epitope. This is ABC prep. I think we will have a demo in Friday. Okay, Friday. Okay. So yeah, I'm not intent to go to each of the websites. So we wait for Friday, so we can do a little bit of tutorial and demo on how we use this website to predict epitope. Right? Yeah, then submit your data and click submit. Then you get the epitopes. Okay, prediction of linear epitopes. This one is for the linear one, remember? So we didn't discuss the conformational one yet. And this one is just B cells. We are yet to go to discuss the T cell. Okay, so for linear epitopes, it's easily predicted computationally, easily identified experimentally. Immunodominant epitopes in many cases do not need 3D structural information because they are linear, easy to produce and check binding activity experiment. But the cons, only 10% of the epitopes can be classified as linear. So actually there are more conformational epitopes compared to linear epitopes. So overall, about the sequence-based prediction methods to predict the linear epitopes, low the problems is analogs to the problems of representing the surface of the Earth on the two-dimensional map. The reduction of the dimension lead to distortion of scales, direction, and distance. So it's just like complicated word. So a simpler word is you need to imagine protein. It is like a 3D structure, right? World map, like our world, it's sphere. So when you want to analyze this Earth in the form of linear, it's can be inaccurate. Okay. Yeah, just imagine a protein sequence is a linear string of amino acid. So when you predict from that linear string, it's inaccurate because the actual protein structure is a 3D shape. Okay. Therefore, predicting the location and composition of epitopes within the protein sequence, it's like it's like studying the spherical Earth in linear. Okay? So what is more sophisticated is using a 3-dimensional structure to pathogen protein, analyze the structure to find surface exposed regions, right? So you have you really need to identify the epitope from the 3D structure. So previously you submit sequence. When you use the tools, you submit the sequence of amino acid. But for this one, you need to submit the structural information. So you can usually get this 3D protein structure from protein data bank. Have you ever done that? So you just download the 3D structure from PDB and then submit it to this platform to predict the epitope. But of course, the PDB or protein data bank need to have the protein structure first. If no researchers have ever done the structure of the protein, so yeah, you need to do it from scratch. You need to identify the protein structure by yourself first. Okay, X-ray crystallography again, and then site-directed mutagenesis. Okay, so this is experimental method to identify conformational epitopes, and this one is prediction method. Okay, based on the data experimentally established to find common sequence or structure method. Develop computer algorithm that can predict if a given anonymous protein sequence has similar method. Okay, this is the protein structure prediction method. This is prediction method using computer. So it's either homology modeling or trading or ab initio. Do you have any courses on protein structure prediction?
It's we have in the
So yeah, it's similar. What we are currently doing is to finalize the structure of the protein, but we focus on the epitope, to finding the epitope. So this is protein structure. Maybe you can try to relate with your other lecture. Okay. Right. So I just go to these examples of software that predict epitopes based on the structure that you submit. Okay, we have this code top. Okay, so this code top here. See, they ask you the PDB code and the chain. So it's based on the structure of the protein or LEPRO. Okay. So we'll try to do a demo with this website on Friday. Or maybe other website. I hope that this website is still working because it's lecture slides from 2 years ago. Okay. Okay, and the second type of epitope, so we have covered all the B cell epitopes. So you can predict epitopes by submitting the sequence of amino acid or by submitting the structure, the 3D structure of the protein. Okay, for linear as well as for confirmation. So T cell epitopes is actually just the same concept, but now it's related more with MHs. Okay, never mind. First we'll discuss about the antigen presentation pathways. Yeah, so this is why you need to do some revision on immunology that we have discussed earlier. So so for the T cell route, you need to have the cells that present the antigen to immune cell, either T helper or T cytotoxic. Okay. Sorry. So actually my department currently meeting, so it's department meeting. There are some issues with my agenda. Okay. Oh, that is sorted. So we'll continue with this. Okay, I already forgot about that. We continue with this antigen presentation pathways. Okay. So for antigen presentation pathway, there are two pathways: endogenous and exogenous. So endogenous is it's like viral infection or tumor that the antigen go inside the cells. So that's why we call it endogenous. So the bacteria or the virus or its own tumor cells that go inside the cell, intracellular. That's meanwhile exogenous pathway, it's like bacteria or parasite, they don't enter the cell, they are in the bloodstream, whatever. So that's why we call it exo, it's outside. So the antigen is outside. Meanwhile, for the endogenous, like viral, that viral infect our cell, they will be inside, right? Our cell. So that's why we call it endo, it's inside. Okay. For endogenous pathway, this is any target cells, because like COVID, they attack your lung cell. So just imagine this is your lung cell. Okay. The viral particle will enter your cell, right? And then your cells will destroy that viral particle using ubiquitin proteasomal pathway, and then the peptide will be brought by what we call this ER, endoplasmic reticulum, to the surface of the cell and will be picked up by MHC class 1, major histocompatibility complex class 1. Okay. And this peptide will be recognized by cytotoxic, by killer cell, cytotoxic killer cell, and they will just kill your cell because it's already infected by a virus. Okay. Meanwhile, for the second part is exogenous pathway. So the antigen, some part of it or all of it, the bacteria will be injured, will be eaten or phagocytosis by antigen presenting cells, for example, dendritic cell, macrophage. So that's all APC, antigen presenting cells. And then the antigen presenting cells, the phagosome inside the cell will lyse the peptide, right? And then go into lysosome. The lysosome will bring the peptide, phagolysosome, into the surface and will be picked by MHC class 2. Right? So MHC class 2, and this will be recognized by T helper cell. So this T helper cell, they will go back to B cells for antibody development. Okay. Right, so this is like this. From proteins to immunology. Oh, okay. So proteins is here. The exogenous antigen or the endogenous antigen, the whole protein that enter the cells. So we call it the whole protein. So this means from proteins to immunogen, from the whole protein that enter the cell out of all protein, only a small fraction, 20%, are processed into peptides. Only 20% are processed into peptide by ubiquitin pathway or by lysosome. by the vegetable cell, okay, and then only a very small fraction of this peptide, from this peptide, only 0.5% bind to the MHC molecules, which are essential for presenting the peptide to T cells. And finally, only the small fraction, like 50% from 0.5%, peptides will actually elicit a response from cytotoxic T lymphocyte. So from this figure, the overall probability of any given peptide being immunogenic is very low. So like from the whole peptide, so only a very small fraction can actually elicit immune response. Right. So this is roughly about the MHC that we have discussed just now. It is polymorphic. You can find MHC actually in human we call it HLA, so human leukocyte antigen. So MHC is very polymorphic. Okay. Class 1 is class 1 bind to shorter peptides because their peptide is from viral just now, viral effect. Viral entry, and the viral will be degraded by ubiquitin or proteasomal degradation, so their peptide is shorter. It's around 8 to 10 amino acid length, and their end here, down here, is closed end. Meanwhile, for the MHC class 2, there are no ubiquitin or proteasomal degradation in MHC class 2, so the peptide will just be phagocytosized by lysosome, and usually without ubiquitin degradation, the peptide is longer, so it's more than 13 amino acid length, and the end of this MHC class 2 is usually open end. Certain amino acid residues here in the peptide. So this is your MHC, or it has MHC. Certain of it, we call it anchor residue, anchor position. It is critical for binding to MHC molecules. So this anchor residue fit into a specific pockets, right? We call this pocket or groove. So what the website or software is predicted is this groove or pocket, the binding site, the binding groove. Okay. So there are two types of binding groove. MHC class 1 molecules interact with the end terminal, the beginning of the groove, as well as the C terminal, the end of the groove, and thereby leaving a bulge in the middle. So you will have a bulge in the middle of the binding. This restricts the length of peptide. So that's why the length of the peptide that can be accommodated by MHC class 1 is just around 8 to 11. Yeah? Because the binding groove is just at the beginning and at the end. So in the middle, they just like bulging there. Meanwhile, for MHC class 2, it can accommodate a longer peptide because of the structure of their binding route, which have a lot of what we call anchor residue. They have a lot of anchor residue, so they can accommodate longer length of peptide. Okay, cannot see my own slide. Okay, so it's a polymorphism. This slide is about the polymorphism of MHC molecules. Right? The MHC molecules or HLA exhibit a high degree of polymorphism. The MHC molecules of humans are referred to as human leukocyte antigen. MHC class 1 are encoded by gene present at 6 loci, HLA-A, B, C, E, F, G on chromosome number 6 of human. Meanwhile, class 2 HLA-DV, HLA-DQ, DR, DM, and DO. Okay, the HLA alleles are highly polymorphic, so it can goes up as high as 3,000 allele for HLA-B. So this is just information about the MHC molecules for polymorphism. They need to be many shapes of MHC because there will be a lot of antigen, so they need their polymorphism are needed to be specific to certain antigen. Okay, right next is quiz. Just skip this quiz. Okay, again, this is a method, so it's actually just the same. It's just like a platform. What I discussed previously is just the principle of how you get the result, the prediction result from this platform. So, like B cell epitopes prediction, T cell epitopes prediction also have various algorithm or approach on how they predict the T cell epitopes. Okay. So remember, T cell epitopes is the epitopes that bind to the MHC. So we have a few here, amphifaticity profiles of the protein. So this one refers to this one, the platform will analyze the distribution of hydrophobic and hydrophilic amino acid along the sequence of the protein, MHC protein. Okay. The reason is amphipathic regions are those that contain both hydrophilic and hydrophobic and hydrophilic, and their profile can provide insight into the protein structure and function. Second one is sequence motif. The third one is quantitative matrix. This is, I think, the same with B cells. It's just a mathematical model. Fourth one is artificial neural network. It's a computational models that take information from the structure and function of protein. I actually tak tahu sangat or can I give you assignment on these topics? Methods to predict T cells epitopes. Susah, sorry. Susah is difficult, or cheating is difficult. Can I give you assignment on this? In this property, yes. Introduction. Yeah, let's get introduction about the MHC complex. Then focus. I'll give the title later today on the website, but just discuss. The assignment is on the different method, different approach and algorithm to predict the T cell. Okay. It's time now. So class should end by what? But I already tired. Okay. So yeah. Okay. I have the title for the assignment now. So this one I will put a nice sentence on your e-learn. Okay. So I'll skip these two slides. So this is also will be covered during demo, but yeah, we'll do it together. We'll look at your website, your platform together on Friday. This is ProPrep 1. I don't know how to pronounce this. So I have to check whether the tools still existing or not. And this net MHC something. Okay, this is inclusive on your assignment. Okay, this is how the algorithm calculate There's four for predicting epitopes for T cells. Okay. So you know that every algorithm, usually they have this four blah 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 and how they predict. Okay. So we just move to the design. I think this is nearly the end of the slides. This is just, okay, we have discussed all these tools, epitope prediction tools. So this one is back to the application of the immunoinformatics. So previously in the first earlier slides, we discussed that informatics can be used or can be applied as diagnostic tools in vaccine development as well as immunotherapy. Okay. Diagnostic tools, for example, the use of computational methods to predict the antigenic targets of pathogen, which can then be used to design a specific diagnostic test. For example, in the case of COVID-19, immunoinformatics tools were used to predict the protein sequence that are specific to the SARS-CoV-2 virus. Okay. So this information was then used to design specific diagnostic tests such as PCR assay, the RT-PCR kit, that can detect the presence of the virus in patient samples. So your RT-PCR kits, whatever, use your saliva samples saliva samples, and then they do RT-PCR. So previously, before they perform the RT-PCR, they need to determine what sequence they are looking for. Okay, so that sequence must be really specific to only the SARS-CoV-2. The second one is immunoinformatics for vaccine development. There are more than 400 microbial agents that are associated with disease. However, vaccines are not to say limited, but not as much as existing pathogen or antigen. So this is like 5 years ago. So vaccine for only 36 pathogens are developed. So yeah, what I've heard, like pneumococcal vaccine, hepatitis, cervical cancer, what is that? HPV vaccine. And then what is BCG for? Toxoid, tetanus, toxoid, so it's very little. But and then the death due to infectious and parasitic diseases make up 19% of total death in the world. But I also have to agree at some point that I think I don't know, I'm not anti-vaccine, but I think human no need to be vaccinated against all antigen or all parasite, because we also need to train our own immune system to be capable of fighting or expel this infectious or parasitic pathogen by themselves, or if we are very dependent on vaccine, we will be a very weak species, I think. Right, this is vaccinomics. Vaccinomics is just development of vaccine using all the omics data or approach. You know, omics, all this genomics, proteomics. So currently, for developing a vaccine, scientists just looking at all this omics data, by data mining and bioinformatics from genome sequence to vaccines. Okay, So this is roughly a categories of vaccine. We can have live vaccine that is able to replicate in the host but are attenuated. They do not cause disease. Some of the vaccine is actually a real virus or real pathogen, but in the weakened form. Subunit vaccine, part of organisms. Genetic vaccine, like foreign DNA. Epitope vaccine, recognized portion of pathogen, like malaria, RTS, and T vaccine. So from the epitope prediction, just now you do the epitope prediction, you can develop a vaccine that can recognize that epitope. Okay. So epitope-based vaccine can be more potent, can be controlled better, can target multiple conserved epitopes in rapidly mutating pathogens like HIV and H3V, can induce subdominant epitopes, and can overcome safety concerns associated with entire organisms or proteins. Okay, so this is current vaccine design. Yeah, you need to go to the lab, do or have blah 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 blah, clone and express purify protein, animal study, try to vaccinate animal, and then do immunoassay, identify antigen, develop vaccine, which the highlight is just to show that it's a very long process to develop vaccine using conventional approach. So that's why we are currently using bioinformatics approach to develop new vaccine. So since we have already a lot of sequence, gene sequence, protein sequence database, and then we can just study the disease-related protein from protein database, and then we can just predict the epitope from the disease-related protein. Okay, when we get the candidate of epitope, we can do the vaccine, right? Either peptide or multi-epitope vaccine. This is vaccinology. Conventional method of vaccinology where include microorganism to be cultivated. So it's usually the development of this vaccine usually will take around 5 to 15 years to develop the vaccine. 5 to 15 years of experimental or of studying before antigen selection and validation, and then the vaccine is developed. So overall maybe take up to 20 years. Reverse vaccinology. So actually the conventional vaccinology, it relies more on empirical, we call it empirical approach, a very conventional or traditional approach, because the vaccine are developed based on a try and error process. Yeah, you don't have any previous information, so you just try and error. So it involves inactivation of pathogen or weakening the pathogen or using the whole pathogen vaccine. And then the second one is reverse vaccinology. It involves the use of bioinformatics and genomics to identify potential vaccine targets by analyzing the genome of the pathogen. So this approach was first started for bacterial vaccine, particularly both organisms with complex or poorly understood biology. Okay. So this will take around 1 to 2 years. And the last one is structural vaccinology, the latest method. It combines structural biology techniques such as X-ray, crystallography, with immunological approach to understand the molecular interaction between pathogen and immune cell. So structural vaccinology, they enable the rational design of vaccine. We call it rational design of target epitopes. Okay. What is actually a rational design? Okay. Actually, rational vaccine design is an approach to vaccine development that use detailed knowledge of the immune system, the structure of the pathogen, and how they interact with the immune system. So instead of relying on trial and error methods or conventional just now, rational vaccine design employs a systematic understanding of the biology and immunology of both the pathogen and the host. So rational means you have already predicted the epitope, for example. Compared to traditional, it's not rational because it's trial and error. You don't have any previous information about that particular pathogen or that particular species. Oh, so the last one, this is the last slide, I think. Okay, yeah. So the last one is, so previous one, what we have discussed here is the immune epitope prediction database. So this is overall immunological database, development and management of various types of database that store information on the immune system, including data on antigen, antibodies, epitopes, immune receptors, immune cells, and immunological pathway. So this is a very important if you are developing vaccine or if you are studying immunology, whatever topics, whatever you're trying to do. But I think if you are an immunology person, this website or this database is very important, the IEDB, Immune Epitope Database. The first one is a database of epitopes and other immune relevance data, including assays, molecular structures, and literature references. The second one is IMGT/HLA database. This is a database of HLA sequence and related data, including allele frequency, disease association, and immunogenetics. The third one is VDJ database. This is a database of curated TCR and VCR sequence and associated metadata, including antigen specificity and disease relevance. The third one is Allergen Online. Okay, this is a database of allergen sequence and information, including IgE binding epitopes and clinical relevance. And the last one is PHI, the database of molecular interaction between pathogens and their host, including interactions with immune cells and molecules. So if you ever work with immunology and you want to incorporate bioinformatics in your study, so this is the website that you must take as your Bible, right? Oh, so that is the last slide, and I think we finished early, 12:30. Is there any question on this topic? So actually, immunoinformatics is just a very simple topic. I don't know why it gives me like 2 hours. It's just an explanation of what immunology is and how currently, nowadays, scientists use bioinformatics to incorporate into immunology studies, so we call it immunoinformatics. The most prominent examples is epitope prediction for vaccine development. So that's right. Thank you so much. How long do you want me to give you for your assignment? So habis betul. Well, we should eventually finish. So today is 24 April. 1 month, is it enough? Or longer? When is your exam week? Oh, you have no exam?
Yeah, only December.
Oh, okay. When will the... So let me check. Usually I also have a deadline for submitting your mark. So, okay. Let me check your timetable. It will end today, 5 July. Okay, I give you until like second week of June. Okay. second week of June. Okay, boleh? Boleh is can. So because you are 100% coursework, yeah, must be a lot of assignment. So like second week of June, 14 June, okay? Deadline. Assignment. Okay, alright. So around 5 pages. Fine. 5 pages. Okay, thank you very much. It's nice meeting you guys. So we'll see you again on Friday. Friday we'll do, yeah, how many hours? Is it 2 hours again? Friday? I also don't know what to do for 2 hours. We'll just play around with the website. Okay. We'll see you again Friday. Bye.
Thank you.
This is long
بس مجموعة موسيقى كانت جدا عظيمة شكلها بالعرض كانت جالية تستخدم بس ليش؟ لأن كل الوصفات كانت جالية للبديل هاي في المنطق بس هاي الوصف يقول مش مثل سمي لا يقول معي قال لي كتاب روتين ومكتوب عليه ريفيو والله ما راح نكتب خطة بعد سنة اليوم كانوا الناس كلها خطة بعد بس انا بكتب بس زين بس انا صار عندي معلومات كافية عن الموضوع. طيب ليش ما انطتنا عنوان مال السايمون مال 4 ورقات 5 ورقات إذا راح أخلي يعلمنا 5 ورقات تقول عن notes. They're better notes. Notes. Sure?
Yeah.
Okay. Good luck. ChatGPT PDF. ChatGPT PDF. This is there. ChatGPT. This one.
Yeah.
Try about it.
知道了，你可以现在知道了。好，我们决定了，我们期中就是去热浪岛了。对，你要不要去？
我期中玩。
啊，我去到13号到15号，你要不要去？
我说我期中去台湾。
哦，是哦。大叔要去
你们几个人？
啊？
你们几个人
5个吧，5个。他应该会去。我叫大叔帮你叔叔也会去。都1000多块的，最好团。Right, we'll send the details later. Traveling. Yeah. 哎，你还有课？跟你的老师做了一个对子。
怎么样？
还好，还可以跟我90分。
啊？我以为是那个呀。
那个推而已。不是我没有跟他讲，那个推是我随便做的，2个小时做完的东西。我都没有啊，我都比较认真做，我都还没有找到我要找的那个数据库。
下一次
去热浪，
热浪岛给我，还有下午上课呢，现在没放了，没放了，没放了。他们快到1点的时候就没有唱，都是2个放。你今天去的时候肯定有点，他们再快一点，1点的话就难拉了就是。欢迎好，是啥？欢迎好，都行。
他走了，去参加。
哦。
关于他的那个毕业设计图。
他们有，还有师兄师姐。
晚。
企业内部。
find the ratio. Like you're considering you said you're going to work, right? You're going to boiling water. So if you're gonna boiling water, then how are you gonna extract the essential? Oh, you're not gonna extract the essential, just boil it. Okay, boil it. You're not gonna use the treatment animal or something. It's just called high treatment. Good. I thought you were gonna extract oil from the flower and then you can extract it. I don't see that. Oh, so you're just gonna, I don't know what like. So I was wondering, that
我们其中去热浪岛一个去，对啊，热浪岛在 KK 啊。
我们带这个，
哈
我们带这个，
，KK 有在那个 KK 有。
Maybe two.
还没还。
5 to 12.
No lighter.
No, I went to them.
Yeah, but no lighter.
I went to them every Monday.
No lighter.
I went wishing to over this with Brian. So much to do with her this year. And she's older than me by 1 month only. Okay, what's yours for this year?
Good. 不能加。要熟练这个手法。Yes, yes. Take video again, take video again. Yes.
You want me to do it again for you?
No, do it. You wish already?
Yeah. Oh my god.
You have 3 wish, you know?
Oh. He wants one.
If I have 3 wish, the first wish I give. Can I have 3 more million wish? No, the first and second one you can say, and the third one you keep it to yourself.
Now we can say that. Just say it again.
Happy birthday. Congrats for getting old.
You're not saying. Who? I do remember doing what did you do? How come you're doing that? I don't know. I just don't. Yeah, you can do that. Well, what I think that's
看现在是不想这个。
Where you like to stay?
yeah. You really looked at
You should try the